Hi guys, you got the notes for this like 2 classes ago. It has the exact same title on your sheet as it does on the screen, so go ahead and find that. If you are not here on that day, then I've left a few extra copies with Miss Wells so you can see her for a copy if you're missing one. So in this unit, we are going to be talking about blood, and it's going to be in 2 formats. So we're going to talk about blood as it deals with genetics and blood types, and then we'll talk about blood as it deals with blood spatter and blood patterns, and those will be 2 separate sets of notes. So today's unit, we're talking about serology. We'll talk a little bit about blood history. We'll talk about how you get the blood types you have and talk a little bit about the genetics for that, and then we will apply blood to a crime scene, like how you would actually process that type of material and what kind of information you could get from it. So starting off with a broad-based definition, so I'm going to underline, which means that you should be highlighting or underlining for yourselves. Forensic serology is the detection and classification, and it's also the study of bodily fluids. And those fluids can include, but are not limited to, blood, semen, fecal matter, sweat, and we look at how they relate to a crime. So what information we can pull from this type of fluid found at a crime and how that can help us to solve it. There are cases where your serologist may also be involved in DNA analysis, and they may also work alongside the spatter pattern analysts, so the people that analyze blood stain patterns. And that should be pattern, not patter. So make sure you write in an N right there. So when we think of the material blood, we always think that it's this red liquid that we have in our bodies. Blood is actually a tissue. So, whoops, sorry. Let's try that again. So blood is a tissue. Somewhere off to the side, like at the side of your slide, I want you to write that a tissue is a group of cells that perform a similar function. So again, a tissue is a group of cells that perform the same function. Okay, so write that off to the side of your slide somewhere. And blood is actually made up of 2 different kinds of material. So we have solid parts to our blood, and that's going to be your red blood cells, your white blood cells, and your platelets. Your red blood cells help with transport of nutrients and oxygen and that kind of thing. Your white blood cells are part of your immune system. So it helps to prevent diseases, or it helps to protect you, I should say, from diseases. And platelets help you to clot. So when you get a cut and you form a scab, that material that makes up your scab, some of that material are platelets. They're just little broken up pieces of cells. So that's the solid part of blood. Then we also have a liquid part of blood, which is your plasma. Now the red color of blood comes from the red blood cells. This plasma tends to be a pale yellow color. Okay, and generally speaking, blood is responsible for transportation of things around your body, including proteins, hormones, clotting factors, nutrients, and also oxygen. Okay, so this is the only history that you need to know for the blood unit, and as usual, we're going to divide it based on the date, who did it, and what they did, so that's what you're going to need to know. And make sure, actually this is not going to be its own separate test because it's only 5 of them, so I will roll that in to our test over blood itself. So again, pay attention to date, who, and what they did. So starting in the 1900s, this man named Castle came up with a test to determine whether or not a red substance was actually blood, and we still use it to this day. It's called the Kastle-Meyer test. and it's a presumptive test for blood. Presumptive just means that it tells you it is blood or it isn't blood. Then in 1901, Karl Landsteiner discovered 3 of the 4 blood groups or blood types that we still use today, and that would be he discovered A, B, and O respectively. In 1902, De Castello discovered the 4th blood type, which is AB, and again we still use all 4 of these to this day. Then in 1940, Karl Landsteiner discovered the Rh protein, and this determines whether or not your blood is positive or negative, so if you're A plus or B minus. And then in 1971, Dr. Bloomberg developed, a method for using antibodies, which are a type of protein in your system, to detect things. So he found a way to take proteins that our body naturally makes, and he uses those proteins to detect or to determine if something is present in our blood. So our immune system, which is made up of a bunch of cells, has to have a way to tell the difference between the cells that your body makes naturally for itself and the cells that are invading your body and could potentially make you sick. So we refer to this as the ability to tell the difference between self versus non-self. The way your body does that is it sticks little proteins on the surface of every cell that it makes, and those proteins identify that cell as being a part of your body. So if your white blood cells come up or come upon a cell that doesn't have those proteins, then it knows that this particular cell was not made by my body. I can go ahead and attack it. So having the presence or absence of certain proteins will determine what blood type a person has. So this part right here, that's important. Now for blood types, these particular proteins are called antigens, or they're sometimes called cell surface proteins. Same thing. And again, either having them or not having them is going to determine what blood type you are. So when it comes to blood types, there are 2 major antigens. Whoops, sorry. Okay, and those 2 antigens are called antigen A and antigen B. Those are going to be the proteins that sit on the surface of your red blood cells. So what blood type you have depends on how these proteins play together. If you have antigen A protein, then you have type A blood. If you have antigen B protein, then you have type B blood. There are some people that have both type A and type B on the same blood cell, so those people have type AB blood. And then there are people that have neither A nor B, so those people have type O blood. Okay, so that's how it goes. If you have antigen A, type A blood. Antigen B, type B blood. Antigen A and B, type AB blood. And antigen A nor antigen B, type O blood. The other part of your blood type is made up of a symbol, and that symbol is either going to be positive or negative. Whether you are positive or negative depends on whether or not you have the Rh factor. That's another kind of protein. So Rh factors determine if your blood type is going to be positive or negative. About 85% of people have Rh plus, and everyone else, so that's 15%, they have Rh negative. Okay, and again, if you are plus, it's because you have the Rh. If you are minus, it's because you do not have the Rh. If we broke down the blood types in the United States, like in terms of the population, this is what it would look like. So I need you to know that the most common blood type is type O, with 43% of people having that. And that the least common type is type AB, with only 3% having that. Okay, it's really good that we have it distributed this way because type O blood has some specific properties, which just means that it can be given to any of the other blood types without causing diseases or without causing clumping or issues when those two blood types mix together. Right, so a lot of the vocabulary is going to sound similar, especially because most of it has the word anti in it, but if you just bear with me, hopefully you'll get it. So the proteins on your red blood cells are called antigens, but your white blood cells are capable of producing proteins as well, and some of them produce a protein called an antibody. So antibodies are part of your immune system, and it's their job to mark cells that need to be destroyed and also to cause them to clump together so that they can't escape. Like, think about it that way. So let me write that for you somewhere. All right, so hopefully you can see what I've just added to the slide. It's right here. You can write that wherever you have some space, but I just need you to know that antibodies are proteins that mark cells for destruction, and it causes them to clump together. So white blood cells can make a bunch of different kinds of antibodies, but some of the ones that they make are called antibody A and antibody B. Antibody A will cause clumping when it comes into contact with antigen A, and antibody B clumps when it comes into contact with antigen B. So establish that in your head because that's something that you need to know. So again, antibody A clumps with antigen A, antibody B clumps with antigen B. So this ability of your white blood cells to make antibody A and B could potentially pose a problem because people who have blood type A have antigen A, and people who have blood type B have antigen B. So if their white blood cells make the wrong antibody, it could cause the blood in that person's body to clump, and that would not be a good thing. So to avoid that, your body is really smart, and it does the opposite. So I need you to know the following things that I'm about to say. So make sure you're underlining or highlighting as we go. People that have type A blood actually make antibody B, because that would mean no clumping. People with type B blood make antibody A. People with type AB have both of those antigens, so it means that they don't make antibody A or antibody B. And people with type O have none of the antigens, so they make both antibody A and antibody B. And when your body follows this system, it makes sure that your blood won't clump with the antibodies that you naturally produce. So it prevents the problem before we even start. All right, so this diagram is just to give you a visual to help with the blood types and which antibodies are made. So here we have blood type A. Notice that this red blood cell has A proteins all on its surface, and that's why that person has type A blood. Notice that their antibodies, which are these purple markers down at the bottom, they have the B type, which means that there's no clumping happening between A and B. For type B blood, there are B proteins all on the surface of the red blood cell. The person makes antibody A, but with their white blood cell, so again, there's no clumping between the two. This blood cell has both A and B proteins on its surface, but it produces neither antibody A nor antibody B, so that there's no clumping. And then here is type O blood, where there's nothing on the cell surface, so there's no A or B proteins because they don't have any. So that means that they make both antibody A and antibody B, so again, there is no clumping. So in this slide, we are going to go over how we perform blood tests, and I'll explain the clumping versus no clumping thing in pictorial form. Hopefully that'll make it make a little bit more sense. So whenever we give transfusions or deal with blood in general, it's always good to know the blood type of the person that we are working on. And blood typing tests are the way that we identify that blood type. So we take a little sample of your blood, 2 or 3 drops, and we perform 3 separate tests, one on each blood drop. And depending on the reaction that we see, it is going to tell us what blood type you have, so both the letter and the plus or minus sign. Now in this particular example, we're just looking at the letters. And I've been using the terms clumping and no clumping a lot. So I want to point out what that looks like. So I'm going to draw some arrows in. So there, and here, and here, and here. Do you see how it looks like some of the color has like joined together and it's all like close to each other, all touching? That's what clumping is. And clumping shows that you are positive for something, that you have that particular thing. Now look at the other two pictures. The only two pictures I have not drawn arrows to, so I'm going to circle those in black. That would be this one and this one. Here you can kind of see a change in color, but there's no, it doesn't look like a bunch of the particles are moving closer together like it does in the other pictures. So here it's for the most part the same consistency. Here it's the same consistency. This would be no clumping. Then here, 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 and here, those are all examples of clumping. So on this particular test, wherever you see clumping, it means that you have that thing. We're going to react all of these blood drops, with two different proteins, an anti-B protein and an anti-A protein, and we can also react it with an RH protein. It doesn't show it in the picture, but we do that in real life, and that'll tell us if you're positive or negative. So right here, this blood group did not react to antibody B, so that means that this person does not produce the B antigen. Right here, it did react to antibody A, so it means that this person produces the A antigen. This person has type A blood. So again, wherever you see clumping, that's what you have. So there's clumping under A, so they have type A blood. Right here, there's clumping for B, they have type B blood. In this one on the bottom, there's clumping in both sections, so that person has AB blood. Okay, so quick little genetics lesson. So the ABO gene, which is the gene that determines your blood type, is what we call an autosomal gene. And autosomal just means that this particular gene is not on a sex chromosome. It's not on chromosome 23, it's not on an X or on a Y. Instead, it's found on chromosomes 1 through 22. One of those are the body chromosomes or the autosomal chromosomes. As a matter of fact, the ABO gene is actually found on chromosome 9. So I need you to know that it is autosomal and that it's found on chromosome 9. Now I need you to think about the words dominant versus recessive. So freshman year of biology, you talked about that a whole lot when you did genetics. And we probably talked about your dominant gene being your stronger gene and your recessive gene being your weaker gene. So let's establish which ones are strong genetically right now. Blood groups A and B are dominant, so they're stronger than O. Okay, so again, blood groups A and B are dominant over O. They're only dominant over O. Blood groups A and B are co-dominant to each other. Okay, so if dominant means strong, co-dominant means that they are both strong. They are equally strong. Because they are equally strong, an individual person can have both A proteins and B proteins, which is why we have people that have AB blood. Okay, now every person gets 2 copies of genes from their parents for their blood group. You get 2 copies of every gene. 1 copy comes from your mom and 1 copy comes from your dad. How those genes interact and play with each other determines what your blood type is going to be. All right, so here I gave you a working definition for some of those genetics terms that we used, and a lot of these you already know. Just make sure you know them. So dominant means stronger, but in the case of genetics, it means this is your gene that's going to be expressed. This is the one we're going to see. And it's always represented by a capital letter. Recessive means that this is the gene that is not expressed. So by itself, it's not expressed. You're going to need two of them in order for it to be expressed. And it's normally represented by a lowercase letter. In the case of blood groups, we use the letter O, and that's the only way that we get O blood. And note that it says it can only be seen if both genes are recessive. So think of it as being slightly weaker to the point where you need 2 times as much for you to be able to see it. Codominant means that one gene is not stronger or does not overpower the other. They're both expressed. I can't get to that with my pen, but whatever. Okay, so in terms of actual blood groups, in terms of the genes that lead to blood types or blood groups, there are a couple of ways you can have A blood and B blood. You can be homozygous dominant, which means that you have 2 uppercase letters, or you can be heterozygous, which means you have 1 uppercase letter and 1 lowercase letter. But remember, in terms of blood groups, we sometimes use the letter O to represent the recessive trait. Okay, then to be recessive, you have to be OO, so that person would have O blood. And to be codominant, you can be AB, which means that person will have AB blood. So I'll show you what I mean. So let's say we're going to draw some Punnett squares. Let's say we have a man and his wife, and the guy is big A for his blood type, and let's say mom is big A and O for her blood type. So they can have children that have A blood, but there are 2 ways that they can have A blood. They can have big A, or they can have big A and O. But because O is recessive, it means that the person's going to have A blood because that's the stronger one. That's the one that gets expressed or shown. Okay? It's the same thing for B. Like if we did A and O, like let's say dad is A and O and mom is also A and O, then kids can be A blood. Okay, and they could be big A, or big A and O, or they can have O blood because you have 2 O's coming together, and that's the only way you can have O blood. All right, so let's go through and fill out this big Punnett square together and just talk about all the possible combinations for blood types. So we're going to write the letters on top, and then we'll write the blood type kind of in the bottom. So this, this person would be A, so their blood type would be A. This person would be AB, so their type would be AB. This person would be AO, so their type would be A. Then this person would be AB. And realize it doesn't matter if it's AB or if it's BA, it's the same thing. So their type would be AB. This person would be BB, so their type would be B. Okay, this person would be BO, so their type would be B. Then over here we would have AO, so their type would be A. Then we would have BO, which gives them type B. And then this last person would be OO, and their type would be O. Okay, so let's talk a little bit about how we utilize blood evidence from the scene of a crime. Now, there are 3 things that we're going to start off our investigation with once we found blood evidence. And they are going to be, first of all, is the substance actually blood? Because there's lots of things that are red. Then we're going to ask ourselves, is this blood from a human source or is it from an animal source? And then third, if it is a human source, we're going to try and identify who this blood belonged to. Could it be one of our suspects? Is it our victim? What's going on? In terms of the evidence that blood gives us, a lot of the evidence is going to be class-based, and that's because a lot of people and things share some of these common characteristics. So it's going to be difficult to highlight pinpoint a particular individual. So some of the things that we can figure out is what species does the organism belong to. So that could be human, it could be rabbit, it could be dog, but it's not going to pinpoint a human. It's just going to tell us, oh, this sample A, that was human blood. Then we can figure out what blood type it is. So that could be AB, or O. But everyone in the world shares those same 4 blood types. So I could narrow it down to a group of people. Like, for example, my blood sample might be AB blood, which means that I'm looking at people that have AB blood, but I'm not going to be able to tell that it came from this particular person. Then we could talk about the rhesus factor. Is this a positive blood type or a negative blood type? But again, everyone in the world is either positive or negative. We can also look at things like, are there diseases present in the blood? Can we tell that this person is sick? And while that might really help to narrow it down, it's going to be difficult to just get the one person with just this one disease in the whole wide world happen to commit this crime. Again, even diseases are things that we as humans will have in common. The only way we can really get individual evidence out of blood is by running a DNA analysis on that blood sample. So when we get to the scene of the crime, we are going to attempt to answer those 3 questions. Now, they may not all be answered at the crime scene, but over a period of time, they should be all answered. The very first thing that we do once we've found the sample and collected it, we can do a quick little test to determine if it's actually blood or not right there at the crime scene, but sometimes we'll send it back to the lab. It just depends on how much time we have. So the first test is called the confirmation test, or the presumptive test for blood, and it involves a color change reaction. Now, there are 2 completely different tests that we can do. Test number 1 is called the Kastle-Meyer test, and test number 2 is called the leukomalachite green test. You don't have to do both. You only have to choose one because they both give you the same information. Both of these tests is just going to tell you if this sample is actually blood or if it's ketchup or beet juice or Kool-Aid or whatever. It actually won't tell you all those things. It'll just say, yes, this is blood, or no, it's not blood. And we figure that out by the change in color. So for the Kastle-Meyer test, okay, I'm not drawing very well today, guys, but you'll deal. So for the Kastle-Meyer test, you're looking for a pink color change. If the blood solution turns pink, then yes, you are working with actual blood. For the leukomalachite green test, just like the name suggests, you are looking for a green color change. If it turns green, then that sample is blood. Once you've determined if you're working with blood, you then want to figure out if the blood is from an animal or if it's from a human. And the way we do that is by running what's called the ELISA test. Now, this has to be run in a lab. And the ELISA test has multiple steps to it, and I will actually walk you through it on the other 2 slides because there are pictures there and it's a little bit easier to understand with the pictures. Okay, so before you start making fun of my bunny, I had to draw this by hand, and you know I don't draw well, so I thought I did pretty good, all considering. So this is the ELISA test, and this is pretty much how we would go about it. We would choose an organism. It's normally a small one, so a rabbit or a mouse works really well for this. We will take a known sample of human blood, and we will inject it into our animal, our rabbit. So before we move on, let's take a little look over here on this side of the slide. So human blood cells, as you know, have these little proteins on them, and those are going to be your A or B proteins. And you know that your immune system is going to make the opposite antibody for whichever protein we have. So we inject our blood into the rabbit. the rabbit's immune system is going to recognize that these blood cells don't belong to the rabbit, they belong to something else, and it's going to send its white blood cells to get rid of this foreign invader. So the rabbit's white blood cells are going to make antibodies, and those antibodies are going to be able to neutralize or prevent the human blood from moving around, and it's going to do so by binding to it or by causing it to clump. Okay, so this is called making anti-serum. The whole reason we do this is because we want these antibodies. Okay, so we're going to inject our rabbit with our human blood. We're going to leave it alone for long enough for the rabbit to make antibodies, and then I'll show you on the next slide what we'll do with that. Okay, so once we know the rabbit has made the antibodies, we will then take a sample of blood from the rabbit, and we will pull the red blood cells off and just keep the serum. We will then mix the serum from the rabbit with the blood from our crime scene, and we're looking to see if it produces a band or a precipitate. So this silver band in the middle right here, that represents where the two sets meet, and it causes clumping between the antibodies and the blood cells, and you get this band. The only time you're going to get this band is if the blood is human. If the blood is not human, the two liquids are just going to mix together, and that's that. So serologists don't just work on blood, they also work on other bodily fluids, so we're going to step through the 3 or 4 most common ones. The first being saliva. So saliva is made up mostly of water. There's also a material called mucin, which helps it to be slippery, so it allows you to swallow your food and stuff a whole lot easier. And then there is a special kind of enzyme called amylase that's used for the digestion of starch. Okay, and then we can also have cheek cells known as buccal cells, and that's where we'll get DNA from. So when they talk about taking a throat swab so that they can run DNA, they're not running the DNA from the saliva itself, they're running them from the cells in the saliva. So in order to, well, before we jump into that, we run quite a few saliva kits and saliva tests because it's one of those forms of evidence that we find with assault type cases. And we can even collect saliva from bite marks. So one of the ways you test to make sure that the liquid you're working with is actually saliva is to do a starch test. So you take starch and iodine, you mix it together. Starch causes iodine to turn a really dark blue, almost purple color. So I need you to know this. And then you need to know that amylase breaks down starch. That's what it digests. So if the sample that you're looking at is actually saliva, when you mix in that starch color, which you can see because you've turned it purple, when that color starts to disappear, it means that the amylase has broken down the starch, which is why there's no more color, and that liquid really is saliva. If the color stays put, like it doesn't go anywhere, then you're not working with saliva because it doesn't have any amylase. Okay, another fluid that serologists will work with is semen, which is made up mostly of water, spermatozoa, enzymes, and organic salts. And spermatozoa are just sperm cells, depending on what name your biology teacher used. You may not recognize that. Again, these are going to be sexual assault type cases that we see a lot of this particular kind of fluid in. And we start off by doing presumptive tests, and then sometimes we'll do confirmatory tests. So a presumptive test is just to determine whether or not we could be working with semen. It doesn't tell us if we're not working with semen, but it will tell us that we are, based on if it tests positive or not. So one of the first things that we can do is we can shine a UV light on it because semen will fluoresce. It'll glow, just like how a white shirt glows under a black light, same kind of concept. The other thing we can do is we can test for the presence of a specific enzyme called acid phosphatase. So acid phosphatase is normally part of semen secretions, and you can do a specific test for it, and it'll turn purple to indicate that semen is actually present in that sample. Okay, so if it turns purple, we know we're working with semen. If it glows under a UV light, we know we're working with semen. Another way to confirm for a fact that it really is semen that is made up of that particular sample is to do a quick little microscope examination. We can just put some of the solution on a slide and look at it under the microscope, and we'd be able to see the spermatozoa cells, potentially. And then, of course, we can do DNA typing as well. The last body fluid that serologists will work quite a bit with is urine, and most often we use it to detect the presence of specific kinds of drugs within the body. So this is more for drug detection than anything else. And the method that we use is called EMIT, and it stands for Enzyme Multiplied Immunoassay Technique. And we just create antibodies that bind to certain drugs. So we mix the antibodies with the drugs, and if the drug is present, it'll cause clotting, and you'll be able to see that, and then we'll know that there are certain kinds of drugs in that urine. All right, so at this point we are done with notes. You picked up vocabulary when you walked in. Whatever time you have left in class, you should go ahead and fill out that vocabulary sheet. The majority of those are going to be in the notes that you just took. There are 2 of them. I think agglutinin is one, and antibody antigen response is another. You're going to have to look those 2 up, but everything else is in your notes. Yep, so that's that. I will see you guys next week. Take care. Bye.